Hi, Professor and classmates. I'm Jina Park, studying in the Plant Genomics Lab under the Professor Kang. Today, I'll talk about my research, phylogenetic analysis using the chloroplast genome data. First, I'll explain of my research about phylogenetic analysis, and then I'll talk about the method and skills. The taxonomy of organisms has been mostly classified according to the traditional classification. Plants are classified based on physiological features or common morphological features such as vascular or non-vascular, seed or non-seed. Compared to a large number of traits of plants, the more the number of correlated traits, the deeper the phylogenetic relationship. However, with the development of next-generation sequencing, whole genome sequencing has become possible, and phylogenetic analysis using molecular information is made. This allows traditional taxonomy and phylogenetic relationship to be verified and modified. Molecular information includes ribosome, mitochondria, and chloroplast DNA. Plants mainly use the chloroplast DNA. Then, why chloroplast genome? Chloroplast is one of the organelles present in the cells of plants. Chloroplast has its own genome DNA, and genotypes are passed from one parent type to all offspring. It is different from nuclear DNA inheritance, and the DNA sequence does not change well because it grows independently. And nucleotide substitution rate is relatively slow. In other words, chloroplast genomes are highly conserved over generations. For this reason, chloroplast genome is a good material for evolutionary research. The comparison of the complete chloroplast genome provides useful information for reconstruction of taxonomic locations and for verification of phylogenetic relationships. Also can find taxonomic locations for new species. Then I'll explain the method and skills I used in my research. First, I use Python as a programming language for processing or analyzing biological data, such as FASTA, GenBank. Python is a popular programming language in bioinformatics. It has an intuitive structure that is easy to learn and understand compared to other programming languages. BioPython is a set of freely available tools for biological computation written in Python. And Anaconda or Miniconda can make easy to install and manage the Python packages or libraries. Then, where do we collect a lot of data? NCBI GenBank database is open access and annotated collection of all publicly available nucleotide sequences and their protein translations. Now, in NCBI database, there are genome data of about 96 million animal species and 43 million plant species has been uploaded. There are various types of sequence data such as mitochondria, ribosome, and chloroplast. Here I collected chloroplast genome data as a GenBank format with sequences and its annotations. The collected GenBank files are processed. Figure is the Python code for parsing and processing GenBank files. First, only CDS is filtered from the GenBank file and translated into a protein sequence, and genes of various species are merged in one data frame to create FASTA file for each gene. Then, alignment is performed for each gene. Frank is multiple alignment tool for DNA, codon, and amino acid sequence. Its phylogeny-aware alignment algorithm has been shown to produce good alignment for evolutionary inference. It's based on a novel algorithm that treats insertions correctly and avoids overestimation of the number of deletion events. In addition, Frank borrows ideas from maximum likelihood method used in phylogenetics and correctly takes into account the evolutionary distances between sequences. It can be run with command line. Models of protein evolution or amino acid replacement describe the probabilities of change from one amino acid to another. Therefore, models are necessary tools for evolutionary analysis. Protest is a Java program to find the best model of amino acid replacement for a given protein alignment. Given a protein alignment and a tree topology, the program calculates the likelihood under each candidate model and estimates model parameters. BEAST is framework for parameter estimation and hypothesis testing of evolutionary models. It is Bayesian statistical framework used combination of prior knowledge and data information. It is very difficult to understand algorithm, but to explain the Bayesian theory briefly, uh, after an event occurs, the posterior probability of the cause of the event is obtained using prior information that is already known before the event occurs. Also, BEAST is cross-platform for Bayesian MCMC analysis of molecular sequence and is Java platform and open source. And let's see workflow of BEAST. First, align the sequence, in my case using Frank. Then generate XML in Beauty. Beauty is a GUI application in the BEAST package. Here, set parameters and best fit model, in my case using Protest. Then, after running BEAST, analyzing BEAST output. This is graphical user interface of Beauty and set parameters and model. Then running BEAST. Then after running BEAST, analyzing and visualizing BEAST output. This is the phylogenetic tree from tree file. Here we can check the phylogenetic relationship between the species and the taxonomic position and check if they are well supported through posterior values. This is all of my phylogenetic analysis. In addition, we perform the other works. For example, to obtain our own data, we also conduct experiments such as DNA or RNA extraction. Also, by using machine learning such as random forest, it is possible to select SMP that can distinguish species. This is all of my present, and thank you for listening.